In the world of wellness, you've probably heard of beta-glucan. It gets talked about a lot. It's this so-called super compound you find in things like oats and mushrooms. But if you look past all the hype, there's a really serious scientific story that's been unfolding for decades. And it puts this totally natural molecule right in the middle of the incredibly complex fight against cancer. Okay, let's get into it. So you gotta wonder, right? It's a frustrating question. We've got this amazingly sophisticated defense network built into our bodies, but somehow it can still be outsmarted. The simple truth is that even a perfectly healthy, robust immune system often just can't keep rapid growth of cancer cells. The natural response is there, but it's often not strong enough to win that battle all on its own. All right, so to really get to the bottom of this, here's how we're going to break it down. First, we'll talk about the core problem cancer presents to our immunity. Then, we'll introduce beta-glucan, not as a cure, but as a kind of trainer for our immune cells. After that, we'll look at the incredible evidence from mouse studies, and then, well, the more complicated story when it comes to humans. And this part's crucial. We'll see why not all glucans are the same. And finally, we'll look at what the future might hold for all this. So let's start with the battleground itself. See, our immune system is constantly on patrol, and it really can recognize tumor cells as foreign invaders. But just recognizing them, that's only the first step. This right here, this is the fundamental challenge. Our bodies produce antibodies that are supposed to tag cancer cells for destruction, but the army of immune cells that's meant to destroy those tagged targets, it often just lacks the firepower. It's kind of like having fantastic intelligence that tells you where the enemy is, but not having enough troops to actually act on it. And that gap? That's exactly what scientists have been trying to fill for years. And that brings us to beta-glucan. Now, this is not a cure, like chemotherapy. That's not how it works. Instead, decades of research have shown it's what they call a biological response modifier. So think of it less like a weapon and more like a drill sergeant for your immune system. So what even is this stuff? It's basically a complex sugar molecule, a polysaccharide. And it comes from really common sources, the yeast that makes bread rise, certain mushrooms, even your morning bowl of oatmeal. This natural origin is, for sure, a huge part of its appeal. Okay, so how does this drill sergeant actually work? Well, it's this brilliant three-step collaboration. First, your body does its job. Antibodies go out, find and latch on to cancer cells. Then, beta-glucan steps in. It doesn't attack the cancer directly, no. It primes your key immune troops. We're talking about cells like macrophages and natural killer cells. This priming is like giving them a critical software update. And finally, these newly supercharged immune cells can now effectively see those antibody tags on the cancer cells and go in for the kill. Beta-glucan basically connects the dots. Now for the evidence. And this is where the story gets really, really compelling and also a little complicated. We have to start with the preclinical animal models, the mouse studies, because the results there have been, frankly, nothing short of remarkable. So here's the central tension of our whole story. What we see in laboratory mouse models is often dramatic. It's clear-cut. But translating that incredible success into human patients with all of our genetic diversity and complexity, that's one of the biggest challenges in all of medicine. And understanding this gap is absolutely key to understanding the beta-glucan story. And this isn't some new health fad that just popped up. Scientists have been on this trail for over 60 years. The very first studies linking yeast components to tumor effects in mice, those date all the way back to the 60s. By 1983, a specific mushroom glucan was already approved as a cancer therapy drug in Japan. This research has been a long, slow, steady build. I mean, just look at one of the most eye-popping results from the lab. There was this 2009 mouse study by a team led by Driscoll. They combined standard antibody therapy with a highly purified particulate beta-glucan from baker's yeast. The result in mice with really aggressive tumors? 80% long-term survival. A number like that makes everyone in the field stop and pay very close attention. So with most data that strong, what happens when we try to replicate it in human clinical trials? Well, this is where we have to manage our expectations a little bit. The story becomes more nuanced, for sure, but it is still very promising. Looking across a bunch of human trials, a really clear pattern starts to emerge. Beta-glucan is almost always used as an adjunct. That means a helper, alongside standard treatments like chemo. For advanced lung cancer, an IV yeast glucan increased response rates. For colorectal cancer, it nearly doubled them.
For esophageal cancer, a mushroom-derived glucan was shown to enhance treatment efficacy. So the pattern isn't a standalone cure, it's one of improvement.
Let's just visualize one of those results. This chart is showing data from a human trial in advanced colorectal cancer. Okay, so standard care alone got a response in about 16% of patients. But when they added an IV yeast-derived glucan to the mix, that response rate jumped to 30%. I mean, that is a significant real-world improvement for people who are very sick.
And this is so important. In human trials, the benefits often go way beyond just shrinking tumors. One of the most consistent things we see is that beta-glucan helps patients tolerate chemotherapy better. It can reduce horrible side effects like painful mouth sores and diarrhea, and it can help keep white blood cell counts up. This improves a patient's quality of life and, critically, helps them stay on their primary treatment schedule. That's a huge deal.
And here is the reality check. This quote from a recent scientific review just captures the whole challenge perfectly. While we see these clear benefits in response rates and quality of life, that dramatic survival extension that we saw in mice hasn't consistently translated to humans, at least not yet. And that brings us to the critical question: why? Well, the answer, in large part, lies right here. It turns out that just saying beta-glucan is like saying car. I mean, are we talking about a high-performance race car or a family minivan? The details matter immensely. This quote from two of the leading researchers in the field really just says it all. The source of the glucan, how pure it is, its molecular structure, all of these things dramatically change how it's going to interact with your immune system. This list of variables is everything. Is the glucan from yeast, or a mushroom, or oats? How is it purified? What's the specific shape of the molecule? Is it a big particle, or is it dissolved in a liquid? Each one of these factors can be the difference between a highly active immune trainer and a totally inert, useless substance. Let's go back to that 2009 mouse study for the perfect example. Remember, when they used that highly purified particulate glucan from yeast, they got 80% survival. But when they swapped it out for various mushroom-derived glucans in the exact same experiment, the survival rate plummeted to just 30–40%. Same study, same cancer, just a different glucan source, and a dramatically different outcome. So where does all this leave us? Well, researchers are now intensely focused on closing that gap between the incredible, explosive results in the lab and the more modest but still really important results in humans. So the main takeaway is this: beta-glucan is not hype. It is a legitimate immunomodulator that shows real promise as a supportive or adjuvant therapy. It helps standard treatments work better and helps patients tolerate them. The holy grail now is to figure out the exact formula to unlock its full potential in people. And the path forward? It's all about precision. Researchers are working to optimize the purity and formulation of these compounds, figure out the absolute best delivery method for specific cancers, and learn the perfect time to give it to a patient during their treatment cycle. It's about turning what's been a blunt instrument into a surgical tool. And all of that leaves us with a final, really tantalizing question. As we get better and better at understanding which glucan does what and which patients are most likely to respond, could this humble, natural compound become a key part of a personalized, integrative approach to cancer care? The research suggests the potential is absolutely there.